Hello Arts Magnet Biology students, let's get ready for your photosynthesis and respiration test tomorrow. So your balanced equation for photosynthesis is 6CO2 plus 6H2O in the presence of sunlight and chlorophyll gives C6H12O6 plus 6O2. So you're putting carbon dioxide and water in and you are getting sugar and oxygen out. So that all starts with the light reactions of photosynthesis, and there are 6 steps. The 6 steps start with photosystem 2. There are electrons in photosystem 2, which is an area that is full of pigments and proteins. Sunlight strikes that and those electrons absorb sunlight energy. They get excited and they leave. They head out to the ETC, the electron transport chain. Photosystem 2 has to replace those 2 electrons, and it does so by splitting a water molecule. Splitting water gives you H+ hydrogen ions and gives you O2, oxygen gas, and it also gives you those 2 electrons. In step 3 of the light reactions, we're going to take those negatively charged electrons, and as they move down the ETC, they make other things negatively charged, so they start attracting a flow of hydrogen ions because positive and negative attract. When those electrons run out of energy, they reach photosystem 1, which is another area that has lots of pigments and lots of proteins. It absorbs sunlight energy, so those electrons get re-energized, excited again, and so they leave. They move on, and they immediately get combined with NADP+ and H+. Both of them you see have positive charges, so they wouldn't naturally go together. That would be like jamming the wrong ends of magnets together. But with those 2 electrons, we can make them go together, and they now form NADPH. In step 6, all of the hydrogen ions that were being used up here in step 3, that flow of hydrogen ions, they are now going to move through an enzyme called ATP synthase, which is in the membranes of the chloroplast, and it moves all of those hydrogen ions from high to low concentration, and that movement generates ATP. So the 2 important things here are circled in red. NADPH and ATP are made, and both of those are needed to perform the Calvin cycle. And then there are pigments also involved in those photosystems. We have chlorophyll A and B, which are green, and then we have carotenoids, which are orange. The reason we have 3 different pigments is because they all absorb different wavelengths of light. So when you use chlorophyll A, chlorophyll B, and carotenoids, you extend into the growing season for plants. They can absorb a lot more different types of light. So that's the first half of photosynthesis. Now we actually have to make sugar out of that, so that's the Calvin cycle. And in your notes, that's a circular diagram to kind of help you remember that it is a cycle. It keeps repeating. But step 1 is carbon fixation, and carbon fixation is going to combine RuBP and carbon dioxide out of the air and force them to become an unstable 6-carbon molecule. Because you're forcing them to do that, and it's not an easy thing to do, we use an enzyme called rubisco. That molecule that was really unstable immediately breaks into 2 molecules of something called 3PG. It's just an unstable intermediate that really doesn't make sugar yet. We need to do more to it. So it goes into phase 2, step 2, reduction, and that's when you use more energy, ATP, and NADPH, and then we're going to change that 3PG molecule into something called G3P. And the reason we do that is because G3P is half of a sugar, and that's what we're trying to make. So in step 3, regeneration, we're going to take some of that G3P and recycle it back up into RuBP so that we can capture more CO2. So we start with RuBP and CO2. We make this, it gets transformed, some of it goes off to be sugar, some of it comes back up into RuBP so that we can restart the cycle all over again and capture even more CO2. Some plants don't do this, and those would be plants that need to save water. So some of them can do the CAM cycle, and the CAM cycle involves plants that only make sugar at night. Good memory trigger here. AM is morning, and that's when you want to eat plants that use the CAM cycle because they're full of sugar. C4 cycle, they make a 4-carbon sugar. C4 means carbon 4. So instead of making the full glucose molecule, they're just making C4, a 4-carbon sugar, which is much simpler and requires a lot less water loss. So all of this has happened inside of the chloroplast, and this is what they look like. You've got two membranes, and then you've got all this green stuff inside. When you're talking about a whole stack of pigment, you're talking about something called the granum or grana. The stroma is the fluid part, and then all of these wrapped up layers of membrane that surrounds the pigment is called the thylakoid membrane, and that's the part where hydrogen ions are moving through ATP synthase, and that's what generates all of the energy. As far as those essay questions, as long as you remember the photosynthesis lab, the spinach leaf lab, and the fact that it took a little while, it depended on the amount of light available, and that they floated because oxygen was produced, you'll be fine there. 6 steps of the light reactions we just went over, and then we talked about chlorophyll A, B, and carotenoids, and the reason that there are so many pigments is to extend that growing season. So let's move on and talk about respiration now. The equation for respiration is just photosynthesis backwards. So instead of starting with carbon dioxide and water and then making glucose, we start with glucose and oxygen. Those get burned up. Carbon dioxide and water are given off, and the most important part is that we're making energy in the form of ATP. So for aerobic respiration, all respiration starts with glycolysis, no matter what. But if oxygen is around, then we can go into the Krebs cycle and the ETC. So glycolysis occurs in the cytosol, that's the fluidy part outside of all of the organelles, and glucose is broken down into pyruvic acid, which is also known as pyruvate. That gives us 2 ATP from breaking glucose in half. That pyruvic acid or pyruvate is now small enough to go into the mitochondria, and there we convert it into acetyl-CoA and then into citric acid. Once it's done that, it is going to give off some byproducts, and those byproducts are NADH, which is just an intermediate molecule, and FADH2. The only reason we need to make those is we need to get these hydrogens, those extra hydrogens off of them, in the ETC. Krebs cycle really isn't that great at making energy, it's only producing 2 more ATP. The ETC is where it's at, that's where most of our energy comes from. So this is occurring inside the membranes of the mitochondria, and all of those H+ ions from the NADH and FADH2 are going to go through ATP synthase, again, that same enzyme, and as they go through ATP synthase, we make heaps, pants loads, crap tons of ATP. In fact, we get 34 ATP out of this step. Everything else related to respiration can only make 2 ATP at a time. ETC, or the electron transport chain, can make 34 ATP. So that's pretty excellent. Grand total, if you go through all the steps with oxygen present, you can make 38 ATP. However, if oxygen is not present, you have to do anaerobic respiration. Remember that prefix AN means opposite of. So the opposite of aerobic respiration is anaerobic, when there's no oxygen. So once again, you always start with glycolysis. That's going to give you 2 ATP. But when there's no oxygen present, cells are either going to do alcoholic fermentation or lactic acid fermentation, and that depends on what you start with. So if you start with a grain or a plant product, you're going to do alcoholic fermentation, and as a result, you get 2 ATP energy, but you also get ethanol and carbon dioxide as byproducts. If you are a meat or dairy product, you undergo lactic acid fermentation, which produces 2 ATP, but also results in a buildup of lactic acid in those cells. And that's why that type of fermentation makes your muscles sore. So why would our cells bother doing this? It's because it's a backup plan. If you run out of oxygen and you still need energy, you've still got this option. Other organisms, things like bacteria and fungi, they will do this almost exclusively, simply because it's less complex and it's a fast source of energy that doesn't require oxygen and doesn't require a lot of the mitochondrial membrane and enzyme stuff that happens in the Krebs cycle and the ETC. So that's all you need to know for the test. Best of luck to you. Please check out the other videos on YouTube, including the one on the light reactions of photosynthesis, if you need extra help.